So before we move to Travenol, you raised this morning the question of the British Pharmacopoeia definition of a suitable donor. If we could have on screen, please, Nick, SHPL 40245_005. This is the British Pharmacopoeia of 1980, Volume 2, and it is this volume that is referred to in some of the data sheets that we looked at this morning. The first reference that I can find to it is from the data sheet in the 1981 to 1982 compendium. The reference, not that we need to bring it up, is ABPI 5036, and it's about cryobulin. That data sheet would have dated from some time in 1980. The definition of the donor is contained, if we can find the fifth page of this electronic document, and it's page 845 of the printed copy. Perhaps if we could go to the second page, and I think it's, yes. For some reason I have a different reference, but yes. This is the entry for albumin. It says albumin is a solution in a water of human albumin containing a low proportion of salt, and this is the relevant passage for our purposes. It is prepared from pooled liquid plasma obtained from blood from human subjects A, who are, as far as can be ascertained by a registered medical practitioner after simple clinical examination and consideration of their medical history, free from disease transmissible by blood transfusion. B, whose blood has been tested with negative results for evidence of syphilitic infection. C, whose blood has been tested with negative results for the presence of hepatitis B antigen by a method not less sensitive than reversed passive hemoglutamation, thank you. And D, the hemoglobin value of whose blood in terms of a cyanomethahemoglobin solution for hemoglobinometry, British Standard 3985, 1966, is no less than 12.5% WV female donors.
Weight to volume.
Weight to volume female donors, or not less than 13.3% weight to volume male donors. So that is the definition to which reference was made in the data sheets. I note that there are previous volumes of the British Pharmacopoeia. We will look into whether the definition of suitable donors changed in those previous volumes.
So this would appear to mean that a suitable donor, there has to be a medical examination by somebody who's actually a doctor, and he sees no sign of a transmissible disease. There's actually been a blood test for syphilis and for hepatitis B antigen, and he's got quite a bit of hemoglobin.
Yes.
Yeah. So this is prepared from plasma, from the blood of the people who have gone through these various different tests.
Yes.
Yes. So one would expect to see some record of these tests and their results in any centre accepting plasma for plasmapheresis.
Yes, one would expect that. It's not expressly stated as a requirement. There's no record-keeping requirement in the Pharmacopoeia, but it's hard to see how the tests can be done effectively without records.
Well, it's one thing to have a definition. It's another to make sure it'll be able to rely upon it.
Yes.
Thank you.
I will turn then now to the next company. And as I say, although this section has been timetabled as being Hyland, Travenol, and Immuno. In the 1970s and the 1980s, Immuno was a separate company from Hyland Travenol, and indeed was a competitor of it. A very brief outline of the company structure for Hyland, Travenol, and Baxter. The US company was Baxter International, Inc., founded in 1931 as the Don Baxter Intravenous Products Company and headquartered in Illinois, near Chicago. We will see later something from the 1975 World in Action documentary, which includes some footage from outside the headquarters in Illinois and an interview with the Senior Vice President for Medical Affairs of Baxter. Travenol Laboratories was founded in or around 1948 or 1949, and it was a specialist pharmaceutical division within Baxter. Hyland Laboratories was acquired by Baxter Inc. this is, in 1952, and Hyland were based in Glendale in California. As a result of this, we often see reference to the Hyland division of Travenol Laboratories, and that is part of the wider Baxter Inc. group. That is the US company. The UK company was incorporated on the 18th of November 1948 as Wallerstein Co. Limited. The name was changed on the 14th of February 1972 to Travenol Laboratories Limited. The name was changed again on the 1st of February 1988 to Baxter Healthcare Limited. The UK company, again, is not a manufacturer, but it is involved in importing and selling US blood products in the UK in the 1970s and the 1980s. For completeness, sir, I point out that Baxter Healthcare Limited spun off the bioscience element of the business to a company called Baxalta UK Limited in 2015. Again, I make no submission on the current company structure or on any liabilities or culpabilities that arise from it. The witness statement of Bo Taras Wahlberg at WITN 3805001 provides further details. For those following the evidence, again, it is helpful to keep in mind that whenever there is a reference to Travenol Limited, then that is to the UK company, as opposed to Travenol Laboratories or Baxter Inc. or Highland, which is a reference to the manufacturers in the United States. The key names for the UK companies are Travenol Laboratories Limited from 1972 to 1988, and then Baxter Healthcare Limited from 1988. The principal product with which we are interested for the Baxter Travenol group is Hemophil, a factor VIII concentrate. And an overview of Hemophil, and indeed of other products, is contained in a draft witness statement prepared on behalf, by and on behalf of Henry Kingdon in 1990. If we could have on screen, please, Cernic, CBLA 5011_005. Dr. Kingdon was at that time the Vice President and General Manager of the Highland Division of the Baxter Healthcare Corporation, having previously been its Medical Director. That information can be found at CBLA 5011_076. He was approached by the legal team representing BPL in the UK HIV litigation to provide a witness statement in those proceedings. The relevant correspondence can be found at BPLL 0016042_021 and CBLA 5069_074. The statement was never finalised, as the litigation settled in December 1990, subject to negotiation over the terms of that settlement. The version which we're going to look at now is that of the 8th of November 1990. We can see the date in the top right-hand corner expressed in the American style. It is a first draft that was prepared by lawyers on which Dr. Kingdon wrote comments for inclusion and information. The other correspondence for references to which I have referred you earlier indicates that those lawyers had met with Dr. Kingdon in order to speak to him and help them to draft up the statement. On the covering note, Dr. Kingdon wrote, and you can see here, I did my editing in blue or black. In blue, these are changes which should be incorporated into your next draft. I've made explanatory comments. And in pink or magenta colour, these are just explanatory and not intended to be incorporated. When he says, thanks, good first draft. So we can see that obviously Dr. Kingdon has had input into considering the draft and was the source of the information that is contained in it, but it is not a finalised draft. It's not always possible, looking at the draft, to identify when Dr. Kingdon is talking about a practice that stood as of 1990 and when he is talking about a practice that was in place at an earlier period. Sometimes it is made clear, sometimes it is not so clear. And as we go through, that is something which is worth keeping in mind. With those caveats in place, the statement provides an overview of Baxter's approach and of the products that it produced. And as a result, we'll start here. Highland Laboratories produced a factor VIII concentrate named Hemophil from at least 1968. That is at paragraph 28. Highland was the first manufacturer to obtain a license for and market a factor VIII concentrate in the United States. Dr. Kingdon recalls from his time as a physician treating a patient with Hemophil in 1968 at the University of Chicago. This is paragraph 28. Highland introduced a factor IX product, Proplex, in August 1970. That's paragraph 32. I take that to mean that it was introduced onto the American market at that time. Hemophil and Proplex were produced from large pools comprised of plasma obtained from paid donors using plasmapheresis. That is at paragraph 12. Dr. Kingdon referred to plasma pools of 15,000 donations per pool, but it is not clear whether the size of pool varied over time. There is some other evidence that we will come to which suggests that they did. At paragraph 22, Dr. Kingdon makes the point that we discussed earlier, that there might be more than one donation from a single donor in that pool. By the 1980s, plasma donations were obtained from plasmapheresis centres owned and operated by Highland, or independent centres that contracted with Highland in compliance with their standards. And I think it's helpful to go to the actual text now to see what Dr. Kingdon says about that. Could we have on screen, please, Cernic, page 5 of CBLA 5011_5, 005. We'll take it from paragraph 12. The industrial manufacturers fractionate plasma collected from plasmapheresis centres around the United States. Highland processes plasma collected from plasmapheresis centres owned and operated by Highland throughout the United States, and, in addition, a number of independent contractors collect and process plasma under contract in compliance with standards set by Highland. Highland also custom fractionates all fresh frozen plasma collected by the American Red Cross under a plasma fractionation contract. He goes on to describe some details of that contract, which I won't trouble you with. The plasmapheresis industry in the United States developed in the mid-1970s.
Where are we now?
Sorry, paragraph 13 we go to.
Yeah.
The plasmapheresis industry in the United States developed in the mid-1970s and is widely used by the manufacturers. Plasmapheresis enables each donor to give up to 32 donations in 8 weeks, as compared to a single donation in 8 weeks by conventional collection methods. In the mid-1970s, the FDA mandated source plasma as a licensed product. In order to obtain a license for the production of source plasma, every collection centre must be inspected annually. Annual inspections are carried out by the FDA, who look closely at all procedures in the collection centres, including donor selection, labelling of donations and testing. Prior to the direction that source plasma be licensed, this is the corrected version, a certain amount of plasma was imported for the production of concentrates. The uncorrected version read, a number of manufacturers in the United States imported plasma from countries such as Haiti and certain countries in Africa. I'm afraid I can't quite make out the next. For use, I think it is.
For use in the production of concentrates. Yeah.
So that is the uncorrected version. The corrected version, as we can see, makes a more generic statement. All importations stopped following the FDA direction that source plasma and the centres producing source plasma be licensed. The FDA would not inspect offshore sites, which meant that imported plasma would not be approved plasma and therefore could no longer be used in the manufacture of blood products. As a result, since approximately 1978, all plasma used for the production of concentrates in the United States has been obtained from donors in the United States. That is a proposition, so that we will test by some other material in due course. Dr. Kington goes on to discuss blood collection at Highland centres. This is paragraph 15. Highland collects plasma by way of plasmapheresis at donor centres owned and operated by Highland throughout the country. In addition, Highland fractionates plasma collected at a number of independent centres who collects plasma under contract with Highland. All donors are paid a small fee per donation. Plasmapheresis donors may make up to 32 donations per 8 weeks, with 2 donations taken per sitting. Plasma collection centres tend to be located near areas where there is likely to be a population willing to spend 2 to 3 hours donating plasma for a small payment. For example, centres are often located close to universities in order to attract the student population, near army bases, and in the lower socio-economic sections of a city. Donor selection is rigorous, and donors are given physical examinations to check for needle tracks and signs of alcohol abuse. Every donation is coded immediately following the donation. The coding information contains the donor's bleed number, the date of the donation, and the donor's social security number. Each bottle of plasma obtained during a bleed is labelled with this information. After each donation, a plasma sample from the donation, labelled in the same manner as the donation, is sent to the Highland Centralised Testing Laboratory, where it is tested for antibodies to HBV, HIV, and ALT. Testing for HIV was introduced in April 1985. ALT testing was introduced at around the same time. The ALT test is a marker for active liver cell injury and is used as a surrogate test for NA and B. ALT testing was introduced following a mandate by the BGA, that's the Bundesgesundheitsamt, the German regulatory authority. Highland sell factor 8 and factor 9 concentrates in Germany and introduced ALT testing in order to comply with the German regulatory requirements. Testing for HCV has not been introduced at Highland donor centres. Testing of all donations is carried out at Highland Centralised Testing Laboratory, with the results sent to both the donor centre and the manufacturing facility in Los Angeles. Highland operates only one testing site, and all plasma collected by or on behalf of Highland in the US is sent to the laboratory for testing. Each donor centre holds all donations until clearance to ship is received from the testing laboratory. Any donation which fails any aspect of the testing procedure is destroyed by the blood centre which collected the donation and who retain records verifying that this has been done. Donors who test positive for HIV are blacklisted and are unable to make any further donations at any Highland centre or any centre affiliated with Highland. Records are computerised, with donors identified by their social security numbers. The turnaround time for testing and the subsequent shipment of all cleared plasma is 7 to 10 days. Shipment to the manufacturing facility may take up to 1 week thereafter, depending on where the plasma is being shipped from. Once the plasma arrives at the plant, the manufacturing process and all subsequent steps, such as quality control procedures and release by the FDA, takes some months, and it may be 6 months to 1 year before a donation given at any of the blood centres appears in the form of a licensed concentrate. All plasma used in the manufacture of factor 8 or factor 9 concentrates is fractionated at the Hyland manufacturing plant in Los Angeles, although in the past some of the concentrate produced for sale in Europe was manufactured at Lezine in Belgium, and the first heat-treated concentrate, Hemophil T, was originally heat-treated in Lezine. Material received at the manufacturing plant is put into quarantine for a period which varies depending on the amount of product in stock. Prior to fractionation, the barcodes on the bottles of plasma are read to determine whether there have been any human shipping errors. Any donations which are picked out during this process are destroyed. In addition, this process removes any donations from donors who have subsequently been identified as seropositive. While the laboratory test will identify seropositive donors, thus enabling the donation, which is the subject of the test, to be destroyed, the barcode reading at the manufacturing plant will identify, with reference to the donor's social security number, any earlier negative donations by that donor which have not been processed. The computer inspection line was introduced in 1984 to 1985. Prior to this, the same procedures were undertaken manually. Removed from the draft by Dr. Kingdon is the following clause: although under the manual procedure, donations from donors who had seroconverted between one donation and another were unable to be removed. That has been struck through by Dr. Kingdon. A couple of further points picked up elsewhere in the statement about donors. At paragraph 34, Dr. Kingdon comments that regardless of the FDA measures that were introduced in the mid-70s, it was his view that fractionators would not have continued to use imported plasma, as much of it was infected by hepatitis B virus, was therefore unable to be used in the manufacture of concentrates. That's at paragraph 34. In terms of the time lag between a test being conducted or a step introduced and that step taking effect in the product which is available, if we could bring up, please, Sir Nick. It's paragraph 138, page 45 of the document. This is specifically about the HIV testing that was introduced in April 1985. What Dr. Kingdon says is, even though testing was introduced by Highland in April 1985, it took approximately 6 months from that time for the manufacturing pipeline to be cleared of untested plasma, as plasma in stock but untested at the time testing was introduced was put through the manufacturing process. As a result, even after the introduction of testing, some concentrates made from untested plasma and with a shelf life of 2 years remained on the market.
Now, the shelf life, is that once the product has been made?
Unfortunately, sir, we have those words, and I can't interpret them any further.
So there's no further help as to whether the 6 months to a year in quarantine before manufacture is in addition to this or not?
Not in this statement. We may be able to find something elsewhere, but I'm afraid that is a question that we would have liked to ask Dr. Kingdon. But
and I suspect another question you may like to ask him, but I'm asking you because you can tell me if there's anything in his statement to say anything about it. The tenor of what he appears to be saying is that if a donation is shown after it has been taken to have come from someone who has a seroconversion, it's removed.
Yes.
At what stage does he say the donation is pooled with other donations?
That I can't answer. I will go back to the statement and see if I can get any further clarity from it.
Because a consequence would be that it might be very difficult to remove one donation without dumping the whole pool.
Yes. I will go back to the statement and see if I can find more precision on that point.
I mean, I suspect you may not have said anything about it at all, but it's might be interesting to know.
Yes. I will turn. Dr. Kingdon says a number of other things in his statement, and I'm sure so that you will consider the whole statement in due course. He expresses opinions about how Highland's system of checking donors compared with the voluntary donors in the United States, and he makes a number of other points as well. And I will leave that to another day. And I will turn instead to his account of the development of heat treatment for factor concentrates and the difficulties involved in it, and it's at paragraph 36 and following of the statement. I'm not going to ask for all of this to be shown. I will summarise some of it and I will read other parts. Dr. Kingdon joined Highland in 1981, a point which is of general relevance to his statement as well. So before 1981, he is not speaking from personal experience or knowledge. When he joined Highland in 1981, a heat-treated version of Hemophil, called Hemophil T, had already been developed and was awaiting clinical trials. That's at paragraph 45. The product was heated in a dried state at 60 degrees for 72 hours. That's paragraph 54. And it had been developed with the intention of inactivating hepatitis B virus. That is at paragraph 52. 1981 is significantly earlier than the period of heat treatment that we were talking about with Immuno.
Yes. When in 81 did he join Highland?
April 1981.
So this is
paragraph 45.
Before April 81, Hemophil T was already developed.
Yes, and awaiting clinical trials. One of the clinical trials involved saw a preparation of Hemophil T from a 300 donor pool, which had undergone extensive screening for hepatitis B surface antigen, anti-hepatitis B surface antigen, anti-hepatitis B core antigen, and it had been screened for ALT levels as well. Half of the pool was spiked with hepatitis B virus prior to it being freeze-dried and then put through the heat treatment process, with the other half left as a control. The trial was reported in August 1984 as having delayed the onset of hepatitis B virus in chimpanzees from 6 weeks to 6 to 9 months, and as of showing no transmission of non-A, non-B hepatitis. That is at paragraph 48 of the statement, and the relevant publication is CBLA 5061 _002, which is Hollinger et al. Reduction in the risk of hepatitis transmission by heat treatment of a human factor VIII concentrate, in the Journal of Infectious Diseases, 1984, volume 150, pages 250 to 262. The authors there commented that the heat treatment method employed, quotes, has the potential for reducing and possibly eliminating the risk of hepatitis transmission by these products. It is important to note in this and indeed in the other studies that the date at which the trial is published is sometime after the initial results are beginning to come through. A clinical trial led by Professor Minucci using haemophil T manufactured from a plasma pool screened only for hepatitis B surface antigen showed that non-A, non-B occurred in 84% of the patients in the trial. The reference is paragraph 49 to 50 in the statement. The results of that clinical trial were published in The Lancet in July 1985. Our reference for that is HSOC 301563. It is Colombo, Minucci et al. Transmission of non-A, non-B hepatitis by heat-treated factor VIII concentrate. The Lancet, 6th of July 1985, volume 2, pages 1 to 4. Again, the point is that although that was published in July 1985, there is evidence that Professor Minucci was already telling people about the results that he was finding of non-A, non-B infections at a much earlier stage. And in another article that he wrote retrospectively, Professor Minucci said that in September 1983, he informed, quotes, a large number of haemophilia treaters that early patients in his trial had developed hepatitis. The reference to that is PRSE 302333. In a memorandum dated the 24th of March 1984, Professor Bloom, Dr. Krask and Dr. Ritzer informed haemophilia centre directors that trials were showing a 63% attack rate of non-A, non-B on first exposure for previously untreated patients. That is CBLA 301831. The point here, sir, is that the initial hopes that came from the laboratory trial that haemophil T might inactivate non-A, non-B hepatitis were proved to be illusory, and that was done through the use of a clinical trial. And information about that clinical trial begins to be shared from about September 1983. But the trial itself is not reported until some time afterwards. Dr. Kingdon, in his statement at paragraph 50, talks about why these different results were achieved, and he attributes the difference to the fact that the laboratory trial was done on a product which was created from a pool which had undergone extensive screening, presumably in order to try to eradicate the possibility that hepatitis B was already in it, because they were going to spike it with hepatitis B to see the effect. And Dr. Kingdon thinks that it is that extensive screening of the pool which meant that when the heat treatment was applied to it, the heat treatment was effective against hepatitis. But when the product was created, as it were, in the usual way, with just hepatitis B surface antigen screened out, then the heat treatment did not prove effective, and that is why non-A, non-B hepatitis was still transmitted. That is Dr. Kingdon's view. I express no comment on it. Haemophil T was, however, according to Dr. Kingdon, found to inactivate HIV. And if we go, please, to page 20, Cernic of the document, it's paragraph 57, Dr. Kingdon says this. In 1984, after it became apparent that Dr. L. Montagnier had developed a test for anti-LAV, We conducted an experiment in conjunction with him to determine whether the virus was heat labile, and whether, if so, haemophil T was AIDS-free. Frozen samples taken from the patients who formed part of the study being carried out by Minucci were sent to Montagna, who performed assays to determine whether any of the patients had HIV. As controls, we used samples taken from patients who were as comparable as possible, similar age, similar treatment intensity, and who had been treated at roughly the same time and over the same period as those used in the haemophil T study. The assays performed by Montagna showed that while haemophil T did not transmit HIV, the other concentrates used in the study did. Montagna's findings, which were reported in The Lancet in February 1985, provided the first evidence that the HIV virus was heat labile. Although I was verbally informed of the results of the assays in September 1984, they were not formally reported in the medical press until February 1985. The Lancet article can be found at SHPL 0000371_036. It's The Lancet, 2nd of February 1985, pages 271 to 272. I note there so that Dr. Kingdon expresses this as the first evidence that HIV was heat labile. I see you shaking your head, and you're on the same page as a Clifford Chance lawyer in the 1980s, because exactly the point that I anticipate you were about to raise was raised in the draft with Dr. Kingdon. If we could go, please, to page 24. The comments in bold from the lawyer, which again concerns this point about it being the first evidence that it was heat labile, is, are you sure this is correct? I understand that Cutter also reported in or around October 1984 that they had conducted studies showing that their product was heat labile. This was referred to in the MMWR on the 26th of October 1984. And Dr. Kingdon's response, which was an internal response not to be put into the statement, was, yes, I am sure that this is correct. And he says, and I quote, the Cutter study was a laboratory study done in collaboration with CDC, and 1, was flawed by moisture in the dried product, and 2, had no clinical proof of HIV kill. So that is the reason why Dr. Kingdon made the comment that he did. Again, the distinction is drawn between a study which is done in laboratory circumstances rather than a study that is done on the product as manufactured.
Yes.
Dr. Kingdon goes on to say that a heat-treated version of Proplex T, was also introduced by Hyland, although it was later found that non-heat-treated Proplex inactivated HIV due to the use of ethanol in the manufacturing process. That's paragraphs 58 to 61. Hyland subsequently moved away from heat-treated products and instead used a solvent-detergent method of viral inactivation. This was licensed from a technique patented by the New York Blood Center. The resulting factor VIII product was haemophil M, and the process was adopted in order to inactivate non-A, non-B hepatitis. That's at paragraph 41. I won't go through any more of Dr. Kingdon's statement now, sir, but I'm sure it is a document to which we will return at some later stage. Turning then to the UK situation, Travenol Limited, the UK company, applied for a UK product license for haemophil on the 3rd of November 1972. The reference for that is DHSC 303741_104. There is, however, evidence that haemophil had been available in the United Kingdom before that licence application. Could we have on screen, please, Cernic SHPL 40276_181. This, as you will see, is a letter from the Department of Health and Social Security to Travenol Laboratories Limited. It is dated the 12th of October 1972, and it's signed from an MC Lort. It is marked for the attention of B.L. Steer. It says this: I am writing with regard to your application for a licence under Part 1 of the Therapeutic Substances Act 1956 to enable you to import haemophil factor 8 and the visit by inspectors of the licensing authority to your premises at Bowles House on the 29th of July. As you are aware, the visit was made as a result of promotional literature relating to haemophil, a copy of which is enclosed, coming into the possession of the licensing authority, which implied that the product was available in the country. The inspectors, on the occasion of their visit, ascertained that a consignment of haemophil had been imported recently, that stocks of haemophil were in your possession, and that quantities had been sold and supplied to a number of doctors. You should know that under Section 3 of the Therapeutic Substance Act 1956, the importation of a substance controlled under Part 1 of the Act is prohibited unless the substance is consigned to a person licensed by the licensing authority to import it. B. Under Section C of the Therapeutic...
Section 6, I think.
Sorry, Section 6 of the Therapeutic Substances Act, it is an offence for a person, 1, to sell or have in his possession for sale a controlled substance, knowing it to have been manufactured or imported in contravention of Part 1 of the Act or the regulations made thereunder. 2, to fail to comply with the provisions of any regulation made under Part 1 of the Act. C. Under Section 45(1) of the Medicines Act, it is an offence for any person to import any medicinal product except in accordance with a product licence, as also is in the possession of that product for the purpose of selling or supplying. D. Under Section 96(5) of the Medicine Act, it is an offence for a commercially interested party to send or deliver to a doctor advertising material relating to a medicinal product without an accompanying data sheet, or in advance of such a data sheet being sent or delivered to doctors. If we go over the page, E, under Section 124(1) of the Medicines Act, where an offence under the Act which is committed by a body corporate is proved to have been committed with the consent and connivance of, or to be attributable to any neglect on the part of, any director, manager, secretary, or similar officer of the body corporate, or any person who is purporting to act in such capacity, he, as well as the body corporate, shall be guilty of that offence and shall be liable to be proceeded against and punished accordingly. The Department takes a most serious view of this matter, but have noted the undertaking given by Mr. Steer on behalf of the company that there would be no further distribution of haemophil without authority. I understand you are in the process of preparing an application for a product licence under the Medicines Act for haemophil, and I should like to be advised as to when you expect to be in a position to submit the application to the licensing authority. Your application for an import licence under the Therapeutic Substances Act has been received and is under consideration. So I won't take you to the Therapeutic Substances Act.
Can you just go back to the heading on the first page, just so we can see what... It comes from the DHSS Medicines Division.
Yes, it
Thank
does.
You.
It is unclear from the documents that the inquiry has reviewed how much haemophil had been provided to and used by doctors in the United Kingdom at that time. The haemophil product licence, the application for the haemophil product licence noted that while no organised clinical trials of the products had been carried out, it had been, and I quote, administered to 2 patients in an emergency situation at St Thomas's Hospital. The results of these applications of haemophil were described as successful treatments, and they were included in the application itself. The reference for that is SHPL 40275_029. The haemophil application was considered by the Committee of the Safety of Medicine Subcommittee on Toxicity in Clinical Trials and the Subcommittee on Biologicals in December 1972 and January 1973, respectively. Dr. Thomas, this is Duncan Thomas, who we came across yesterday as well, prepared a report for the subcommittees. Could we have that on screen, please, Sir Nick? It is DHSC 0105593_006. We can see in the top right corner, report by Dr. Thomas. The subcommittees are listed just underneath that, and the dates of the meetings are given in the box there. It is stated to be an application under the 1968 Act for a product license. If we could turn, please, to page 14 of this document. This is the medical comment that comes from Dr. Thomas. It's a summary of what he has found of the rest of his report. The availability of factor VIII concentrates represents a major advance in the care of patients with classical haemophilia. Such concentrates have enabled corrective orthopaedic procedures to be carried out, and for the first time there is a prospect of domiciliary treatment. The major disadvantage of currently available commercial preparations, such as haemophil, is that they are prepared from very large plasma pools and carry the risk of transmitting hepatitis virus. Highland screened all their donors for hepatitis-associated antigen, which reduces but does not eliminate this risk. However, no attempt is made to disguise the risk of hepatitis, and it may be considered that the decision to use this material could be left to the individual clinician who can balance the potential hazard against the anticipated therapeutic benefit to the patient. It is recommended that a product license be granted. That is Dr. Thomas's comment. The reference to no attempt being made to disguise the risk is a reference to some of the data sheets and packaging that we will come on to later. Dr. Thomas also provided the subcommittee with his summary of a report that had been produced following an inspection of Hyland laboratories in California and a blood bank in San Francisco owned by Hyland. This inspection had taken place in October 1972. Could we have, please, DHSC 0105593_006? So I'm sorry, page 15 of the document. At the bottom right-hand corner, we can see the initials DPT, which are Dr. Thomas's initials. At the top, we can see that it is a summary of the inspection report. I don't have the full report for you, sir. We will continue to see if we can find it. This is the summary. Hyland Laboratories, Costa Mesa, California, were inspected on October the 24th, 1972. In addition, a commercial blood bank in downtown Los Angeles, owned and operated by Hyland, was also inspected. The blood bank performed plasmapheresis on between 200 to 300 people daily. 1,000 millilitres of blood was collected from the donors in 2 steps, and the cells returned. This was done twice weekly on the regular customers, who constituted about 50% of the total number of donors. The donors were all men, mostly middle-aged, and predominantly of Mexican origin. They were euphemistically described to me as, quote, people who need $5, which is the amount they were paid for each donation of blood. From what I saw, they were certainly not affluent, although they could not fairly be described as down-and-out alcoholics. Neither did they seem to be youthful hippies, for the most part. However, the medical screening of the donors was rudimentary. A microhematocrit determination of earlobe capillary blood, blood pressure and temperature, and that was about all. Probably the most important screening carried out was routine testing for hepatitis-associated antigen using Hyland's own counter-electrophoresis kit. This testing is carried out on all blood that is collected, on every visit, and before the plasma leaves the blood bank. Several aspects of the whole operation do not meet TS regulations. Take that to mean therapeutic substances. For example, the transfusion needle is not inserted by a doctor, the donors are not screened for syphilis, more than 420 millilitres of blood is removed at one session, and a hematocrit and not a hemoglobin determination is made. However, it is hoped that the subcommittee will advise the licensing authority of the relevance of some of these requirements in the present context. At the factory, plasma is pooled, and the pools contain plasma from as many as 6,000 donors. The fractionation process is carried out under excellent conditions in a modern, well-equipped plant. The personnel seemed highly competent and well-informed. My only criticism was that the aseptic filling area was small and overcrowded, and they placed too much reliance on laminar flow cabinets. However, a new filling area was due to be built within a matter of weeks. Standardization of the factor VIII concentrate is carried out using a house standard and not the international standard. They promise to mend their ways, but I'm doubtful if they will, unless required to do so. In conclusion, obviously the main problem with this product is the hepatitis hazard. The donors do not inspire confidence, and factor VIII concentrate is prepared from very large plasma pools. Despite the HAA testing, the risk of hepatitis must still be considered to be present. However, the firm make no attempt to disguise this potential hazard. That is the summary of the inspection report, and it was included in the documents that was sent by Dr. Thomas to the subcommittees for their consideration as part of the licence application. Dr. Maycock wrote to Dr. Thomas on the 4th of January 1973. That is the letter that we looked at yesterday, and he expressed the same view and respect of haemophil as he did with cryobuline. The reference is DHSC 013026_163. In short, he generally supported Dr. Thomas's report, but he suggested that initially the licence, if it is to be granted, be restricted to just a few haemophilia centres so that efficacy can be assessed. The problem that we have, sir, is that we have little documentation about what happened next in the licensing process. In particular, we haven't yet identified whether or not any conditions were attached to a license which was granted. The DHSS informed Travenol on the 19th of February 1973 that the licensing authority proposed to grant a product license. A reference for that is DHSC 303741_104. And the license itself was issued on the 10th of September 1973, SHPL 40276_159.
So as from the 19th of February, it was effectively licensed?
Yes.
In the same way as cryoprenin was from the 22nd of March?
Yes. Let me just check that, sir. If we go, please, Cernic, to DHSC 303741, _104. We can see the letter is the 19th of February 1973, and it refers to the application date of the 3rd of November. But then it says, as amended by your letters dated the 3rd and 5th of January and the 5th of February 1973, the licensing authority proposes to grant a product license for, and then haemophil is given, and then there are some conditions attached about the potency of the unit being expressed in international units. The supply of the product will be restricted to hospitals and haemophilia centres, and the provisions set out in regulations 4G, H and I of therapeutic substances, manufacture and importation, general regulations 1963, shall apply. So those are the conditions that are expressly attached there. It goes on in the next page to say the product licence will also include a provision that the number of the licence shall appear on all containers and packaging and the packages in which the product is packed, and on any package inserts or accompanying literature. The formal documents relating to the issue of the licence are being prepared and will be sent to you in due course. Please let me know the date on which the product is introduced onto the market. In terms of cryobilin, there is an express reference to the fact that this letter of intent can be used as a full product licence until the full product licence is received. There's no such expression in this letter. Well,
it seems to almost to follow from the expression, the formal documents relating to the issue of the licence.
Precisely.
So the intention looks to be precisely the same.
I think that is a fair inference from the document, sir.
So as from February of 73, the haemophil was on the market?
Yes.
Officially?
Officially. A further point that I would make there is that there are references to the letters of the 3rd and 5th of January and the 5th of February 1973. We haven't yet been able to identify those. And it is likely, and indeed I'll go as far as probable, that those letters concern some amendments or some undertakings that the company have made in respect of the licensing process in order to get the license through. Unfortunately, we don't have them, or at least we haven't found them yet. The license itself, the formal license, was issued on the 10th of September 1973. That is SHPL 40276_159. And again, as with cryobilin, despite Dr. Maycock's proposal, there wasn't an initial restriction to just a few haemophilia centres. Travenol were also granted an import licence under the Therapeutic Substances Act on the 15th of March 1973 for reference to that as SHPL 40276_167. As we saw yesterday, Travenol were included as one of the parties to the central contract with the DHSS, which ran from the 1st of November 1973. The other party was Serological Products Limited, so it's cryobilin and haemophil, and as we heard yesterday, certainly at one stage the intention was for 5 million international units of each. Travenol Limited submitted an application to vary the product licence for haemophil on the 30th of May 1975, and that is to include a reference to the use of RIA testing for hepatitis B. And it was said as part of that application that each unit of plasma was tested for HBsAg by counter-electrophoresis, and the final product was then tested for HBsAg using the Ausrea Kit 2. Each lot of the product had to have a non-reactive test result before it could be released for sale. The variation application was accepted in June 1975. The reference is SHPL 40276_185. That is 30th of May 1975. We have got to the section of a part of the chronology where the World in Action documentary Blood Money arises. This was broadcast on the 1st of December 1975. The second part was broadcast on the 8th of December 1975, but it is the first part in which Highland's facilities form the basis of the investigation that took place there. It is my intention to show about 20 minutes of that documentary, which shows what was actually happening in and around the donor centres that Highland were using. Are you content for me to do that now, sir?
Do you need time to set it up at all?
It has been set up, so it should...
Well, let's see it now, shall we?
The reference, sir, is MDIA 40113, and it's from about 6 minutes and 4 seconds into...
Valley's now 75 degrees in Orange County and downtown Los Angeles 70, currently in Hollywood 60.
Unlike Britain, where blood is given voluntarily, in America plasma is bought. Much of it is bought from men who need money badly, like those down here on the skid rows of America's big cities. But paid donors carry 6 to 13 times the risk of having hepatitis as volunteer donors, and they can pass it on. To discover why, a World in Action investigator spent 4 weeks visiting plasma centres, selling plasma, talking to donors and examining safeguards. Tonight, World in Action investigates the health risk to Britain's haemophiliacs from the men who sell their blood in America. Blood and plasma products like Factor VIII are big business in America. This is Costa Mesa, California, south of Los Angeles. Here is the headquarters of the Highland division of Travenol, a subsidiary of Baxter Laboratories, a leading US drug company and the makers of Hemofil. The company buys plasma in 13 American states and Puerto Rico. We asked them if we could film inside one of their plasma centers. They refused. One reason, unattractive outside situations and unattractive donors. We therefore decided to investigate for ourselves, starting here in Baltimore on America's East Coast. This is East Baltimore Street, the city's skid row. This area with its bars, sex shops and peep shows is home to many Baltimore alcoholics and down and outs. Here we found the Highland Donor Center. It is 6 a.m. and the temperature is near freezing. The plasma center does not open until 7:30, but the queue of donors starts to form well before then. Many of these men are out of work. Plasma centers are booming because of the current recession. Unemployment is highest among the black population, who make up a large proportion of plasma donors. Government rules say any donor under the influence of alcohol is unsuitable, and centers tell donors not to drink 24 hours before selling plasma. But many we saw did just that. Donors are allowed to sell half a litre of plasma twice a week. They usually get £2.50 for the first half litre and £5 for the second. When the donors left, none of them would agree to be filmed. We decided to continue our investigations in another part of America. Baton Rouge, Louisiana, in the deep south. Across the road from the Salvation Army in a rundown part of town, we found another Highland Plasma Center. Once again, the queue, many of them unemployed, had formed well before the center opened at 8 a.m. The nurse in charge refused to let us film inside. However, we were able to get some scenes of what happens in a plasma center. Plasma is collected by an hour-long process called plasmapheresis. A litre of blood is taken from the donor. This is then spun in a centrifuge and the red cells separated out and returned to the donor. Highland is left with about half a litre of plasma. From 2,000 to 6,000 litres are then pooled and concentrated factor VIII extracted. Hepatitis thrives in unhygienic living conditions such as those often found in warmer southern states like Louisiana. As well as by injection, it's passed on by close personal contact and contaminated food. To assess the health risk from US-paid donors to haemophiliacs in Britain, World in Action invited Professor Ari Zuckerman, a leading British hepatitis expert, to join us. We took him to California, where Highland has 6 plasma centres. California has more cases of hepatitis than any other state.
Well, it's been recognized for a number of years now that raw blood does carry a higher risk. And it's difficult to actually pinpoint the reason, but it seems that individuals who are willing to sell their blood are normally from a background which appears to be rather poor socioeconomically. In the past, many of them were alcoholics, and indeed a well-known dictum which originated in the United States was "ooze for booze." This has recently been replaced by perhaps a more serious element, namely drug addicts.
5 a.m. the next morning, Professor Zuckerman visits the Highland Center at San Jose, 50 miles south of San Francisco. Once again, we find ourselves among men at the bottom of American society. But for the first time, we find a donor who is prepared to talk to us. His name is Gary.
Why did you come down?
I need the money.
Do you have a job?
No,
Why not?
because I can't get employment. I'm on parole.
How much do I get paid? $8. It's my $8 day.
Pardon me?
Do the questions that they ask you in there before you give plasma, do you always answer them truthfully?
Are you going to tell them?
No.
No, you know, it's some, you know. Yeah, most of the time. You know.
But what sort of questions wouldn't you answer truthfully?
Well, they ask you stuff like, you know, if you've been drinking and stuff like that, you know, the night before. You know, if you've been eating right and all that crap, you know. And I'm a big fat dude, so it don't matter if I've been eating right or not. I'm healthy, you know.
Morning, darling. Shove it. Shove it, rapid.
Questioning about a donor's medical history is important in keeping out unsuitable donors. But from talking to donors, World in Action found that it is not unusual for them to lie about their health.
I know what you got. I didn't think I did.
Pardon me while I puke.
By offering to sell plasma in 5 Highland centres, we established even more disturbing facts. 1. No check was made on the false addresses we gave. This can admit down and outs, a high hepatitis risk. 2. Highland doctors did not always carry out the only checks that can detect drug users. Drug users are among the highest risk for hepatitis. 3. Physical examinations were not always done fully, though certified as such. 4. Certain medical questions were not asked, but were filled in as having been answered satisfactorily. The Bureau of Biologics, which controls plasma centres, has also criticised Highland. Since November last year, the company has been warned 13 times for breaking federal regulations at its plasma centres and processing plants. Last year, the Bureau temporarily closed 1 centre. We were asked to stop filming inside the San Jose Centre, but we left Professor Zuckerman inside to observe the donors and what happened to them. While we waited for him, we asked another regular plasma donor, an out-of-work labourer, whether down and outs sold plasma.
Well, mostly it was sometimes, yeah. It's like you're going to the street down and you're broke, got no place to stay. That's the first place you go to. Blood bank. Because otherwise, the guy doesn't know where to do. He's going to look for a blood bank.
Do you know alcoholics who give their blood?
Mostly are alcoholics.
Do you know drug addicts who give their blood?
No, I don't think they'll accept it.
But they accept alcoholics?
Certainly. See, the alcohol, like wine or whiskey, whatever it is, it brings your blood up, puts iron in your blood.
Later, we asked Professor Zuckerman what he thought of the donors he'd seen.
One of the strongest impressions that I obtained is the type of donor that was presenting himself at this center. I was somewhat surprised to see that a number of individuals were clearly malnourished And obviously these are individuals who should not donate blood, and particularly should not have protein removed from their circulation. Another type of person that was presenting himself at the center was the vagabond type, individuals who have just come off buses, people who were ill-kempt, one or two persons who one would probably regard as drug users.
How many of the people that you saw presenting themselves here today would be accepted by the blood transfusion service in England?
Well, this is my own judgment as a physician, would be that most of them would have been rejected straight away.
Professor Zuckerman then took us to meet America's leading campaigner against the paid donor system, Dr. J. Garrett Allen, professor of surgery at Stanford University.
Won't you come on in? Thank you.
The two men discussed Dr. Allen's work on the hepatitis risk from paid donors and the recently discovered extra risk of liver damage from using concentrates. Afterwards, we talked to Dr. Allen.
What sort of evidence is there that shows the degree of risk that you run if you use blood taken from paid donors?
There are a number of studies that have been made in the past decade or less in which the risk runs from 6 to 70 times greater than were the donors all from a volunteer source, friends and relatives and such.
What is the reason for that?
The reason for that is that the paid donor is offered so little money that no one is willing to take time off his work to go give transfusions, and therefore most of these donors are unemployed, are transients, and live in a lifestyle that none of us would put up with.
How effective are the tests in preventing hepatitis virus getting into these pools?
We really don't know how many viruses are involved. There are at least 2 and perhaps more. The major one, hepatitis B virus, is detected fairly well, but it appears that at least 2/3 more infectious bloods or donors will escape detection by the use of this test because the test does not apply to their virus.
We then moved to Los Angeles, where Professor Zuckerman took us to meet 2 other leading experts on the risks attached to factor VIII concentrates, Dr. Alan Redeker and Dr. James Mosley, professors of medicine at the University of Southern California. We asked Dr. Mosley about the risks of pooling thousands of donations in order to make hemophil.
Well, even if you have a very low carrier rate in a population, if it's 1 in 100 and you pool 100 units, that 1 is going to contaminate the other 99. If it's 1 in 1,000 and you pool them, it's going to contaminate the other 999. And particularly if there is a concentration technique which not only concentrates the factor that you're interested in, the clotting factor, but also concentrates the virus. And unfortunately, that happens to be true for the clotting factors. The virus is concentrated along with them. So even with the best donors, a large pool is a risk, and the larger the pool, the higher the risk.
What are the chances of someone catching hepatitis from using a product made from the plasma of these type of people?
If it's a blood product that cannot be sterilized, and that's true for the clotting factor concentrates, the risk is probably 100% if the individual is susceptible.
Dr. Mosley told us many British haemophiliacs could be susceptible, especially those needing few injections. They've had no experience of hepatitis or of products made from large pools of bought plasma. The Highland Donor Center in downtown Los Angeles is in the heart of the Skid Row area. The drinking of alcohol is common around plasma centers. We were told many donors drink to build up the iron in their blood to pass a test before they sell. Others believe it speeds up their circulation, enabling them to give blood faster. But for many men on Skid Row, drinking is an all-day, everyday affair. Here there is no shortage of alcoholics who, because of their lifestyle, are likely to be hepatitis carriers. To continue our investigation into the cause of the hepatitis outbreak in Britain, Professor Zuckerman visited his second Highland donor centre. While he remained inside, we toured the neighbourhood, which has no less than 5 plasma centres and blood banks. Hepatitis is a common disease in the overcrowded, poor areas of big cities like Los Angeles, but it is also a major problem throughout America. At this plasma centre, near Highlands, which is owned by a major independent plasma supplier, we found one man who talked frankly about paid donors and who they are. The manager, Russell S. Tate.
What about alcoholics? Do you think that you have many people who are alcoholics that come in here?
Yes, we probably get a proportionate number. We screen them very carefully, though, and try not to let them donate.
But you still think that you have quite a few that probably get through your screening process?
Yes, I'm sure we do.
What about drug addicts?
There again, that's a possibility that runs fairly high. We try to screen them the best way we can, but it's very difficult to do that.
When in fact you examine the donors, how much do you rely on them answering truthfully the questions that the nurses ask them?
Probably about 50% of our questioning is based on their answers.
And do you think it's safe to rely on a man answering a question truthfully when if he answers the question the wrong way, he won't receive any money, which is really why he's here?
It may not be fair, but it's about the only way we can do it.
Do you think it's ethical for a company to take blood or plasma from people who are on the lowest level of society and then sell it at quite a profitable transaction?
It's American business. I don't know whether it's ethical or not, but up to this point in time, no one has found an acceptable substitute for human blood, so we have to get it somehow.
Back at the Highland Centre, the donors had started to come out. One of the first was Bill, an unemployed dishwasher. He said today was the 176th time he'd sold plasma.
Do you come down here if you've got a job?
No.
So you only come down here
when I'm not working.
Yeah. Have you been working for the last 3 months?
No, I've worked a couple days last week, but not really, not enough to keep going.
Where do you live?
Usually hotels. If I haven't got not working, I stay in one of the missions here in LA.
How much does that cost you?
Hotels run about $2.25. The missions are free.
What in that case, other than if you get $15 a week, is that for you to keep enough to keep alive on?
No.
So what do you do?
Well, I hope I could get work, or if I don't get the work, I'll eat at different missions throughout the rest of the week.
What do you eat at the missions?
Beans, mostly.
Nothing but beans.
Mostly beans. Other things. They have sometimes spaghetti, sometimes, varies at different missions.
Like many Skid Row donors, Bill, who is 33, spends most of his day hanging around the area. Most mornings and afternoons, he comes here, the St. Vincent Centre, known as the Misery House. This is where Skid Row down-and-outs spend their time until the missions open in the evening. Highland, like certain other companies in America making Factor 8 concentrates, relies on hundreds of bills for plasma. But for several years, the company has also bought plasma even cheaper in developing countries. There, the hepatitis risk is often greater. Buying plasma is an international business. Reportedly, Highland has bought plasma in Central America and the Caribbean, South America and Africa. The World Health Organization and the Red Cross are trying to stop the plasma traffic, but Highland still buys plasma for hemophil outside mainland America. Baxter Laboratories, which owns Highland, has become one of America's fastest growing drug companies. Thanks in part to hemophil and other plasma products, since 1970, Baxter's net profits have grown from £7.17 million to £18.14 million last year. After visiting 10 of Baxter's 24 Highland plasma centers, we ended our investigation here in Deerfield, Illinois, near Chicago, at the Baxter headquarters. We put the questions it had raised to Baxter's Senior Vice President for Medical Affairs, Dr. Richard S. Wilbur.
What is your reaction to the cases of hepatitis that have occurred in England amongst haemophiliacs who have been using hemophil?
Reported cases of hepatitis, particularly by Dr. Kraske, which appeared in Lancet last year, occurred from earlier batches which were made before we were able to use the newer techniques of screening for hepatitis B, which we now have. We were very pleased to read that the cases were all mild. I believe that the most severe case, the person was sick for something like 6 weeks. I wouldn't want anybody to have hepatitis, but when one compares the benefit to the patient of taking the factor VIII concentrate as compared with this relatively mild disease, we feel that the risk-benefit ratio for that patient was a good one. But let me emphasize, we have improved our techniques for screening our donors for hepatitis since that date. It is much less likely that future batches will cause hepatitis. We have every intention of going ahead and getting still better techniques for screening for hepatitis.
What's your view of as a doctor, of the quality of the people that sell plasma to Highland?
As a doctor, I don't make any judgments on the quality of people. I accept them all as they are.
What is your view then, not as a doctor, of the donors?
As a human being, I'd prefer not to make that sort of value judgment on the quality of my fellow humans. I find them for what they are.
Have you visited the Highland donor centres?
Only one.
Only one?
Yes.
What did you think of the people that you saw then?
They were human beings, such as I took care of when I practiced medicine.
Can you explain to me how it is, if you're so careful in checking the people that donate plasma or sell plasma, how it is possible that I can go to a Highland donor centre and I can have a physical examination filled out in which questions are put into, to which I have supposedly answered yes, that were never put to me, and examinations are indicated to have been carried out that never were carried out?
No, I can't.
But in fact, can you explain to me why, in fact, if, for example, people are asked whether they're alcoholics, that I have been in Highland donor centres where people have been there and have donated plasma, who smelled of drink, who drank before they came in and went straight out of the donor centre and bought a bottle of wine afterwards?
If you say so, I have no reason to disagree with you.
But I mean, would you dispute that that would happen?
I wouldn't dispute if you say it has.
But doesn't that...
What you've observed, that isn't the protocol we've set up, no.
Doesn't that strike you as disturbing, that we could actually see this?
Yes, we would prefer that all of the plasma were available from better sources. And we do not deliberately seek out as a source of plasma the unfortunate people in the country. As I said before, we would vastly prefer to have it from voluntary donors, just as everyone would like to have blood transfusions from voluntary donors.
How safe do you think paid donors are?
If we could get this factor only from voluntary donors, as you say, from the upper levels of society, we would do that. Until we can, we must get what we may.
Just getting it so I don't get all around.
World in Action's investigation has confirmed that haemophil carries a high risk for 3 reasons: the use of paid donors, its production from large plasma pools, and the inadequacy of hepatitis tests. Next week,
we won't go any further onto the World in Action documentaries. They move back to the United Kingdom after that. So I'm conscious of the time, sir.
Yes, well, we'll take a break, shall we, until quarter to 4.
Thanks.